Hello everyone, welcome to today's lecture on drug targets in drug discovery and development. In the previous lectures, we discussed the overall drug discovery process and how new medicines are developed from laboratory concepts into approved therapies. Today, we will focus on one of the most critical aspects of this entire process, the identification and selection of drug targets. A drug target serves as the molecular destination for a therapeutic agent. Before scientists can design a drug, they must first understand which molecule in the body is responsible for the disease and whether modifying that molecule can produce a beneficial therapeutic effect. The success or failure of a drug development program often depends on the quality of the chosen target. In fact, many drug candidates fail during clinical trials because the selected target does not sufficiently influence the disease process. Therefore, understanding drug targets is essential for anyone studying biotechnology, pharmacology, pharmaceutical sciences, biomedical engineering, or drug discovery. By the end of this lecture, you will be able to define a drug target, classify different types of drug targets, understand target identification and validation, and appreciate the role of modern technologies in discovering new therapeutic targets. Let us begin with the basic definition. A drug target is a biological molecule whose activity can be modified by a drug in order to produce a desired therapeutic effect. In most cases, drug targets are proteins. These proteins may function as receptors, enzymes, ion channels, or transporters. However, advances in molecular biology have expanded the range of targets to include DNA, RNA, and complex cellular pathways. When a drug binds to its target, it either activates, inhibits, blocks, or modulates the biological function of that target. This interaction ultimately changes cellular processes and produces therapeutic benefits. For example, insulin binds to insulin receptors and regulates glucose uptake by cells. Aspirin inhibits cyclooxygenase enzymes and reduces pain and inflammation. Statins inhibit HMG CoA reductase and lower cholesterol levels. These examples demonstrate how drugs exert their effects through specific molecular targets. You might wonder why scientists spend years identifying and validating drug targets before developing medicines. The answer is simple. A drug can only be effective if it interacts with a biologically relevant target. If the selected target is not truly involved in disease progression, even the most potent drug may fail to produce clinical benefits. Drug targets influence drug efficacy, drug safety, side effects, therapeutic selectivity, clinical success rates. Studies have shown that drug candidates directed against genetically validated targets have a much higher probability of reaching market approval compared with targets that lack strong biological evidence. Therefore, target selection is considered one of the most important decisions in pharmaceutical research. Not every molecule in the human body is suitable as a drug target. An ideal drug target should possess several important characteristics. First, it should be strongly associated with the disease process. Second, it should be druggable. In other words, it should contain structural features that allow interaction with therapeutic molecules. Third, it should exhibit sufficient specificity so that modulation of the target affects diseased tissues while minimizing effects on healthy tissues. Fourth, modulation of the target should be safe and should not disrupt essential physiological functions. Finally, scientists should be able to measure target activity using laboratory methods, biomarkers, or clinical endpoints. These characteristics help researchers identify targets that are both therapeutically relevant and practically useful. Drug targets can be classified into several categories. The most common categories include receptors, enzymes, ion channels, transporters, and nucleic acids. Let us discuss each of these in greater detail. Receptors are specialized proteins that receive signals from hormones, neurotransmitters, growth factors, and other signaling molecules. When a signaling molecule binds to a receptor, a cascade of cellular responses is initiated. Because receptors regulate many physiological functions, they represent one of the largest classes of drug targets. Among receptors, G protein coupled receptors, commonly known as GPCRs, are particularly important. In fact, approximately one third of all marketed drugs target GPCRs. Examples include beta blockers acting on beta adrenergic receptors for hypertension, antihistamines acting on histamine receptors for allergic conditions, opioid analgesics acting on opioid receptors for pain management. The widespread involvement of receptors in human physiology makes them highly attractive targets for therapeutic intervention. Another major class of drug targets consists of enzymes. Enzymes catalyze biochemical reactions that are essential for cellular function. In many diseases, specific enzymes become overactive or dysfunctional. By inhibiting these enzymes, drugs can restore normal physiological balance. For example, aspirin inhibits cyclooxygenase enzymes and reduces inflammation. Statins inhibit HMG-CoA reductase and decrease cholesterol synthesis. DPP-4 inhibitors used in type 2 diabetes improve glucose regulation by prolonging the activity of incretin hormones. Because enzymes possess well defined active sites, they are often considered highly druggable targets. Ion channels regulate the movement of ions such as sodium, potassium, calcium, and chloride across cell membranes. These ions play crucial roles in nerve conduction, muscle contraction, and cellular signaling. Calcium channel blockers are commonly used to treat hypertension and cardiovascular disorders. Sodium channel blockers are used in arrhythmias and epilepsy. Transporters represent another important target class. These proteins move molecules across biological membranes. For example, selective serotonin reuptake inhibitors or SSREs inhibit serotonin transporters and are widely used in depression and anxiety disorders. Recent advances in biotechnology have expanded drug discovery beyond proteins. DNA and RNA are now recognized as important therapeutic targets. Several anti cancer drugs act by interacting directly with DNA and interfering with replication. More recently, RNA based therapeutics have emerged as powerful treatment modalities. Messenger RNA vaccines developed during the COVID 19 pandemic demonstrated the tremendous potential of nucleic acid based therapies. Similarly, antisense oligonucleotides and small interfering RNAs can selectively regulate gene expression. These approaches represent a rapidly growing area of pharmaceutical innovation. Once researchers understand a disease, the next challenge is identifying suitable drug targets. This process is known as target identification. Modern scientists utilize multiple approaches including genomics, transcriptomics, proteomics, metabolomics, bioinformatics, and systems biology. Large scale sequencing studies often reveal genes associated with disease susceptibility. Proteomic analyses identify proteins that are overexpressed or dysregulated in diseased tissues. Computational methods help integrate these complex data sets and prioritize promising targets. Human genetic studies are particularly valuable because they provide direct evidence linking specific genes to disease outcomes. Identifying a target is only the beginning. Scientists must then confirm that modifying the target actually influences disease progression. This process is known as target validation. Several experimental approaches are used. Gene knockout models can determine whether removing a gene alters disease characteristics. RNA interference can temporarily suppress gene expression. CRISPR-Cas9 technology enables precise genome editing and functional studies. Researchers may also use small molecules or antibodies to modulate target activity and observe biological responses. A well validated target significantly increases the probability of successful drug development. Even if a target is biologically important, it may not be suitable for drug development. Scientists therefore evaluate target druggability. They ask several key questions. Can the target bind a therapeutic molecule? Does it possess an accessible binding pocket? Can selective modulation be achieved? Is the target accessible within the body? Structural biology techniques such as X ray crystallography, cryo-electron microscopy, and computational modeling provide valuable insights into druggability. The field of drug discovery is undergoing rapid transformation. Artificial intelligence and machine learning are increasingly used to analyze large biological data sets and identify novel therapeutic targets. CRISPR based screening technologies allow researchers to investigate thousands of genes simultaneously. Single cell sequencing reveals cellular heterogeneity that was previously impossible to detect. Advances in structural biology have provided unprecedented views of protein architecture and drug binding sites. Together, These technologies are accelerating the discovery of next generation medicines. Let us examine a successful example of target based drug discovery. Researchers observed that individuals with naturally occurring mutations in the PCSK9 gene exhibited unusually low cholesterol levels and reduced cardiovascular risk. Further investigation revealed that PCSK9 regulates the degradation of LDL receptors. This finding suggested that inhibiting PCSK9 could lower LDL cholesterol. Subsequently, monoclonal antibodies targeting PCSK9 were developed and demonstrated remarkable clinical efficacy. Today, PCSK9 inhibitors are widely used for managing severe hypercholesterolemia. This example highlights the importance of strong genetic and biological evidence during target selection. Despite remarkable progress, significant challenges remain. Human diseases often involve complex biological networks rather than single molecular pathways. Targets may exhibit redundancy, meaning that blocking one pathway may activate compensatory mechanisms. Off target effects can lead to toxicity. Additionally, promising laboratory findings do not always translate into clinical success. Future drug discovery will increasingly rely on precision medicine, systems biology, artificial intelligence, and multi-omics approaches to identify highly effective therapeutic targets. To conclude, drug targets represent the foundation of modern drug discovery and development. A drug target is a biological molecule whose activity can be modified to achieve therapeutic benefits. Common target classes include receptors, enzymes, ion channels, transporters, and nucleic acids. Successful target identification, validation, and drug ability assessment are essential for developing safe and effective medicines. Emerging technologies such as artificial intelligence, CRISPR screening, single cell sequencing, and advanced structural biology are revolutionizing the discovery of new drug targets and shaping the future of precision medicine. Thank you for your attention. In the next lecture, we will discuss target identification and validation strategies in modern drug discovery in greater detail. Thank you and see you in the next session.
Hello friends, let's learn about health. Make sure to like this video and subscribe for more.